Fungus plant pathogen.
Before I begin with plant pathogen fungus, I will introduce the background of fungus. It is a eukaryote that digests food outside and obtains nutrients directly through its cell walls. It has membrane-bound organelles and nuclei but lack of chlorophyll. Fungus can grow in soil, water, or live as symbiotic with hosts. It grows at the tip of hyphae which forms mycelia. Besides, it undergoes sexual and asexual reproduction, which produces mytospores and meiospores respectively. Next, I will talk about the effects of plant pathogen fungi. It has caused harmful impacts to agriculture and plants. Firstly, it lowers the crop yielded and its quality, which causes huge loss in agriculture industry. It is reported that plant diseases have caused 10 to 15% of major crops in this world yearly. Among them, 78 to 80% of crops are caused by pathogen fungi. This impacted the source of income in farming countries heavily. Plant pathogen is called as phytopathogen as well. Phytopathogenic toxins are produced via the metabolism of plant pathogens. Even little amount of the toxins will destroy optimum biological function of plants. They are secondary metabolites that will cause wilting, necrosis, and leaf spotting. Cell death or the death of plant occurs if physiological disorders happen. This is due to the inhibition of making proteins and nucleic acids in the host cells that caused by the toxins. Let me share a fun fact. Corn smut is a plant disease that caused by Ustilago maydis. It's a pathogenic fungus that found in maize plant. It lives as biotrophic. The smut has fungal tissue and host tissue, and this one is called as Villa Coche. It also used as Mexican delicacy. That's all for my introduction. I will let Whitney to talk about the types of fungus pathogen in plant.
Okay, now plant pathogenic fungi can be classified as biotrophic, neutrophic, or hemibiotrophic, which depending on their host interaction for their nutrition. Biotrophic pathogen, which is from intimate interaction with plants and can persist in and utilize living tissues. Necrotrophic pathogen, which will kill the tissue to extract nutrients. And these two ways of the host interaction can be differentiated into biotroph and necrotroph respectively. Besides, the third type is hemibiotrophic pathogens, which start as biotrophs and then switch to become necrotroph.
Now, I would like to give an explanation on the disease cycle of plant pathogenic fungi. Plant pathogenic fungi can live within or on the surface of plants. They will have to adapt to the changing environment in order to survive. Hence, pathogenic fungi generate spores or specific hyphae termed sclerotia to withstand adverse environments. The fungus is ready to infect when host plants becomes accessible in the spring. The fungus generates spores that are carried to the plant by wind, water, or animals such as insects and attached to the host by the secretion of a sticky extracellular matrix. This type of attachment is to keep the spores from being washed away from the host surface before they may penetrate. The infection consists of two stages, which is penetration and colonization of the host tissue. The fungi penetrate the host tissue to obtain nutrients either through natural apertures in the plant or through wounds on the plant's surface caused by insect damage. Hyphae are then generated after penetration process and grow in the tissue during colonization. Some fungi destroy plant cells instantly to feed on their resources, while others draw up nutrients from living cells via a specific structure generated by the hyphae. Let me explain in detail of disease cycle by using the growth of the pathogenic fungi Botrytis bunch rot as an example. As you can see, A shows the general picture of the sclerotia. B and C shows the germination of sclerotia in which hyphae with spores at their tip end starts to form. After that, spores are then released and locate at plant tissues or organs as can see from D to H. At stage I to K, we can generally see that the spores undergo germination, penetration, and invading of the tissue resulting in the formation of hyphae. The plant cells and tissue are destroyed by the pathogenic fungi. Lastly, L shows that new spores are formed when the fungi absorb sufficient nutrients, and this cycle will then be repeated. For information, weather greatly affect the number of disease cycles that are repeated. This indicates why pathogenic fungi undergo different number of disease cycles in a season. Do you know how pathogenic fungi usually penetrate their host? Fungal pathogens usually enter their host through direct penetration of the plant surface. A dome-shaped epidermal cell plays an important role in cell penetration and is differentiated by the plant pathogenic fungi, such as to penetrate their host via the cuticle layer directly. The epidermal cell features a chitin-rich cell wall and an interior layer coated with melanin. In order to overcome the physical barriers presented by the cell surface, adhesion to the plant surface is required, followed by pressure and then enzymatic degradation of the cuticle and cell wall. A series of genes in the pathogen are turned on and off, resulting in the production of enzymes such as cutinase, cellulase, pectinase, and protease, which attack the cuticle, cell wall, and middle lamella during degradation process. To create the pressure required for the hyphae to penetrate the cell wall, the epiderm is first firmly attached to the plant surface with a proteinaceous glue. The turgor-driven mechanical stress on a thin penetration pad ruptures the cuticle at the epidermis base and allowing it to develop into underlying plant epidermal cell. The high turgor pressure exerted, along with some enzymatic breakdown of the host cuticle and cell wall, allow the pad to enter the host tissue. After penetration, colonization comes into place. The pathogenic fungi are known as ectoparasites, which absorbs nutrients via hostoria in the epidermal or mesophyll cells. There are also parasites that enter the host epidermal and mesophyll cells via hostoria by colonizing the space between cuticle and the outer wall. In general speaking, pathogens are able to establish colonies deeper within plant tissues.
A symptoms of plant disease is a visible effect of disease on the plant. The disease symptoms help us to recognize which fungal disease is affecting the plant so that we can apply the right defensive tool to treat the infections. Symptoms may include a detectable change in color, shape, or function of the plant as it responds to the pathogens. Leaf wilting, it's a typical symptoms of verticulum wilt caused by the fungal plant pathogens, which is called verticulum arboretrum. Next, I would like to show some examples of fungal disease sign. It would be leaf rust, stem rust, sclerotinia, which is a white mold on the leaf, and also powdery mildew, as I said just now. For disease symptoms, it would be bird eye spot on berries, damping off of seedling, leaf spots, and also lastly sclerosis, which is the yellowing of leaves. Plants cannot communicate with us, so sometimes it is difficult to understand when they are sick. Furthermore, once disease symptoms on plant tissues, it is impossible to heal the plants. We can only reduce the damage by decrease the spreading of the disease to neighboring. This means that prevention is the key to protection. Prevention is the most effective way to manage plant disease. To prevent fungal disease, there are several methods that we can use. One of the most satisfactory methods would be strict sanitation. It is to eliminate the pathogenic organism, starting with the initial stage of propagation and the growth of potential host plant. Other than strict sanitation, here are some integrated management system or strategies for fungal disease, which include the following. First would be genetically host resistance. Using genetically resistant species, cultivars, varieties, and hybrid. Many of the major crop, cultivars resistance to prevailing disease are available and are more continually being developed by plant breeders. As has been discussed, with other type of disease, if it is available, it should be the first line of defense for disease caused by fungi. For example, Phytophthora infestan. It is a water mold that causes the series of potato and tomato disease known as late blights, or also called as potato blight. Second would be cultural practice. It is recommended to plant only disease-free certified seeds, maintaining a balanced fertility program that avoid excessive or inadequate level of key nutrient. Other than that, effective water management, which include maintain an adequate soil drainage, monitor irrigation practice, and also soil pasteurizations. Thirdly, it would be chemical application, which include the use of fungicides. Very often, the measures are not sufficient to provide complete control of fungal disease. So farmers use fungicides to prevent fungal infections. But please remember that fungicides can only used by the expert under control condition only, because the misuse of this product can be harmful to human, other organisms, and the environment. So, always remember to read and follow the label recommendation before applying it. Lastly, it will be government regulatory measure. It is the implementations of strict quarantines that exclude or restrict the movement of fungal and fungal-like organisms, as well as the infected plant material.
Now, we will show you some examples of fungus plant pathogen. Magna por oryzae, known as rice blast fungus, is a causative agent of rice blast disease. It is responsible for the most serious disease affecting cultivated rice in the world. Alright, let me give you a brief idea about rice blast disease. Rice blast disease is a disease caused by filamentous ascomycin fungus, Magna por oryzae. It is one of the most destructive diseases affecting rice in all rice-growing countries and often cause serious damage to global rice production. This disease occurs in 80 countries on all continents where rice is grown. The extent of damage caused depends on environmental factors, but worldwide is one of the most devastating serious diseases, resulting in losses of 10 to 30% of the global yield of rice. Okay, let us look into the symptoms of this disease. In rice seedling, small necrotic reagents appear initially, which become larger and corrosive and have corrotic margins. In older rice plants, disease symptoms can occur in leaf, junction of the leaf blade, and leaf shade, node, neck, and panicle. Neck root and panicle blasts are particularly devastating, causing up to 80% yield losses in severe epidemics. Triangular purple color lesions form on the neck node which elongate on both sides, seriously impairing grain development. The panicle becomes white when young neck nodes are infected, and the infection later in plant growth results in incomplete grain filling. Next, let us study about the life cycle of Magna por oryzae together. The disease cycle begins with a conidium lens on a rice plant becomes attached to the host surface through production of spore disc mucilage. This is followed by a series of development steps: germination, germ tube growth, formation of an episoriums, emergence of a penetration pack from the episoriums, and subsequently invasive growth in the host. After this pathogen invasive growth in the host, it will enter into the cell death period. In susceptible hosts, the fungus properly spreading from one plant cell to another via plasmodesmata. Besides, the fungus is hemibiotrophic, which is initially feeding on the living cells but subsequently causing cell death. The lesions of the plant surface start to form when host cell death begins 5 to 4 days after infection. Not only that, the spore release is triggered by a 1 to 2 hour period of darkness, and it can sporulate repeatedly for around 20 days. The disease is polycyclic, which means that a multiple infection cycle in a growing season, and it will be around 7 days between spore germination and production of conidia. Lastly, Magna por oryzae. These fungal pathogens not only bring disease to rice growing, it also brings seriously impact to humans. It's because rice provides almost 25% of the calorie consumed by humans. Moreover, the pathogen has now jumped host to web and is now an urgent problem in South America. Therefore, understanding the biology of this pathogen is particularly important for developing disease control strategies.
The other example of a plant fungi pathogen is Botrytis cinerea, which is also widely known as noble rot and causes grey mold disease. It causes significant losses in over 200 agricultural crops globally. The fungus is classified as a typical necrotrophic, infecting the host through programmed cell death pathways. It is most damaging on mature or senescent tissues of dicotyledonous hosts, but it normally acquires access to such tissues much earlier in crop development and remains dormant for a long period of time before quickly rotting tissues, when the environment is favorable and the host physiology changes. The most common symptoms on leaves and soft fruits that caused by Botrytis cinerea are soft rots, which are accompanied by parenchymal tissue collapse and water soaking, followed by the quick formation of grey masses of conidia. On many fruits and vegetables, the infection typically starts on connected senescent blossoms and then spread as a soft rot to harm the nearby developing fruit, to become blossom and rot. The most harm occurs on stems at pruning wounds in greenhouse-grown tomatoes, when the fungus can destroy the whole stem. In blackcurrant, symptomless Botrytis cinerea infection of blossom styles causes premature abscission of developing fruits. Over 50 hosts have been found to have seed-borne infections, including flax, sunflower, and lettuce. Seed transmission in chickpeas can result in yield losses in Australia. Grey mold in this crop usually begins with the rotting of the herbaceous stems at ground level, with various soft rot lesions occurring on leaves and pods. Furthermore, I will introduce the life cycle of this fungus. The crucial survival mechanism for Botrytis cinerea is sclerotia, formed within dying host tissues. However, they vary in size and are not clearly visible in all susceptible crops. In temperate areas, sclerotia begins to develop in springtime to generate conidiophores and multinucleate conidia, acting as a key source of inoculum in a crop. Mycelium also lives within infected dead host tissues left as agricultural waste, as well as within certain seeds, where it serves as major inoculum. In aging cultures, the pathogen also produces many microconidia from phyllates, which serve largely as spermatia. Finally, Botrytis cinerea also brings disease to humans. Despite the high rate of sensitization, Botrytis cinerea has a limited prevalence in both outdoor and indoor settings. Botrytis cinerea causes allergic rhinitis in children and adults, asthma in children, and hypersensitivity pneumonitis in horticultural and viticultural situations. Besides, there is presently no known allergen from Botrytis cinerea.
Thank you, Whitney. Now I'm going to conclude our topics. We know there are 7 types of plant pathogen fungi. They obtain nutrients from hosts by one of the 3 ways, that is biotrophic, necrotrophic, and hemibiotrophic. To reach hosts and begin disease cycle, they form spores and carried by wind or animals to initiate infection on host plant. They go for penetration first, then followed by colonization. Also, adhesion, high pressure, and enzymatic degradation are required as the mechanism of host penetration. The process of self-death is plasmodesmata spread the pathogen from one cell to another, and lesions would happen after 5 days. Sporulation is triggered by darkness and lasts around 3 weeks. In order to control plant disease, what can be done is sanitation, genetic host resistance, cultural practice, pesticides, and regulatory measures. Now we know that fungal pathogens can use different kinds of lifestyles in order to survive in plants. Here comes to the end of our presentation. Thank you.